Da, svi su to, tako da možemo da krenemo.
Dobar dan.
Dobar dan.
Ja sam prvi put ovde, a već imam tako ulogu da vam čitam, što ja to radim po prvi put, bar na tehnološkom fakultetu, tehnološko-metaloškom fakultetu, oprostit ćete mi za greške koje ću sigurno napraviti obilat. Ok, danas smo se skupili ovde, ovo mi deluje kao početak svadbene ceremonije, danas smo se skupili ovde da pratimo i da uživamo u odbrani doktorske disertacije našeg kolege Amita Kumara. Ima tu još nekoliko imena koje ću preskočiti. On je rođen 1992. godine u Indiji, završio je srednju školu u Uttar Pradešu, a završio osnovne studije mašinstva 2014. godine u Laknau, u Indiji, i master studije na politički korektnim, održivim tehnologijama 2019. godine na Indian Institute of Science u Bangaloru. Pod naslovom, i to je veoma važno za nas danas, preliminarna studija o reakciji i analizi pomeranja vodenog toka, vodenog gasa sa hladnom plazmom. To je bio izvanredan uvod u ono što će on kasnije raditi u Belgradu. On je student zajedničkih doktorskih studija na Univerzitetu u Beogradu i Univerzitetu Girona u Španiji u okviru međunarodnog projekta Evropske unije Horizonti 2020, u stvari u okviru Novelties programa, i on je upisao doktorske studije inženjerstvo zaštite životne sredine na Tehnološko-metaloškom fakultetu u Beogradu 2019-2020, to je školska godina, pod rukovodstvom profesora Povrenovića. Od decembra 2019. zaposlen je u laboratoriji za neravnotežne procese i primene plazme Instituta za fiziku, gde je pod rukovodstvom Nevene Puač obavio svoj naučni rad, svoj naučni program, većinu bar, i to je dovelo do današnjeg dana. U skladu sa međunarodnim ugovorom o zajedničkom mentorstvu u izradi doktorske disertacije upisuje doktorske studije na profilu nauka o vodi i tehnologiji vode i na Univerzitetu Girona u Španiji pod mentorstvom Wolfganga Gernjaka. Položio je sve kurseve doktorskih studija, završne ispite na oba univerziteta i uspešno je odbranio temu doktorske teze. Disertacije. Izvinjam se. Evo, prva greška za koju znam.
Odbranit će.
Molim?
Odbranit će.
Odbranit će. Ne, odbranio je temu doktorske disertacije. Amit je do sada autor ili koautor 4 rada u međunarodnim časopisima i u toku je recenziranje još jednog u M21A časopisu. Takođe ima 6 saopštenja na renomiranim međunarodnim konferencijama. Nakon polaganja svih predviđenih ispita, Amir je temu doktorske disertacije prijavio 22.6.2021. Nastavno naučno veće Tehnološko-metaloškog fakulteta na sednici od 29.6.2021. godine imenovalo je komisiju za pripremu izveštaja o podobnosti teme i kandidata Amira Kumara, koja je prihvaćen od strane Veća naučne oblasti tehničkih univerziteta u Beogradu na sednici od 19.1.2022. godine, čime je odobrena izrada doktorske disertacije pod nazivom Razvoj i karakterizacija plazma sistema na atmosferskom pritisku za preradu otpadne vode. Ima i naslov na engleskom. Nakon podnošenja urađene doktorske disertacije, Nastavno naučno veće Tehnološko-metaloškog fakulteta na sednici od 20.9.2022. godine imenovao je komisiju za ocenu doktorske disertacije koja je pripremila referat i koji je bio od 30 dana na uvidu javnosti i na sednici od 29.12.2022. donelo odluku o prihvatanju referata o urađenoj doktorskoj disertaciji i odluku o imenovanju komisije za odbranu disertacije u sastavu dr. Vladimir Pavićević, vanredni profesor Univerziteta u Beogradu, TMF, dr. Ivona Radović, redovni profesor Univerziteta u Beogradu, TMF, dr. Zoran Petrović, član SANU, penzioner, zaposlen kod PIO, dr. Jelena Rađenović, naučni savjetnik Univerziteta u Gironi, dr. Sara Rodriguez Mozaz, viši naučni saradnik Univerziteta u Gironi i Zdenko Mahala, redovni profesor na Univerzitetu Comenius u Bratislavi, Slovačka. Svojim zaključkom od 4.1.2023. Univerzitet u Gironi je dao saglasnost na referat o uređenoj doktorskoj disertaciji kandidata Amita Kumara, kao i na prethodno naveden sastav Komisije za odbranu doktorske disertacije. Veće naučnih oblasti tehničkih nauka Univerziteta u Beogradu na sednici od 23.1.2023. godine donelo odluku kojom se daje saglasnost na referat o urađenoj disertaciji Amita Kumara pod nazivom Razvoj i karakterizacija plazme sistema na atmosferskom pritisku za preradu otpadne vode, čime su se stekli svi zakonski uslovi da se doktorant pozove na javnu odbranu svoje doktorske disertacije. Ova disertacija spada u oblast primene plazme, specifične plazme neravnotežne, koja omogućava da dizajnirate osobine i sastav efluenata, svega čime plazma gađa svoje mete, u ovom slučaju vodu, i na taj način obavite određene zahvate koje ste želeli. U ovom slučaju bavili su se prečišćavanjem otpadnih voda, što je veoma važno u primjeni plazme i u poljoprivredi, a i za ljudsku upotrebu. Neravnotežne plazme imaju tu osobinu da sve poslove obavljaju elektroni, dok je gas, joni i sve ostalo na sobnoj temperaturi, te su imale veliki, kada smo naučili kako da ih pravimo na atmosferskom pritisku, znali smo kako da ih pravimo na niskom pritisku, ali su se ljudi opirali tome da ih vakumirate i da ih ispumpate na 100 militora, i onda smo morali da napravimo plazme na atmosferskom pritisku da bismo postigli primjenu plazme u medicini. Paralelno sa time krenule je i primjena plazme, posebno zato što je naša grupa imala dobru saradnju sa botaničarima, biolozima u našem gradu. Krenula je i ideja o primjeni plazme u poljoprivredi. I tu smo imali dosta uspeha i naša grupa vodi jednu post akciju na tu temu. Amit se izvanredno uklopio u rad grupe i nadam se da će nam efikasno prikazati svoje rezultate, a posle ćemo videti i reći koju reč i o njemu samom. Hvala.
Okay. Thank you, Professor, for the introduction. So, good afternoon, everyone. Welcome to my PhD defense. Today, I'm going to talk about my PhD research, which is focused on design, development, and characterization of atmospheric plasma system for wastewater treatment. Yes. So here is an outline of my presentation. I will go through each points in details. So water pollution due to anthropogenic activities is one of the major environmental issue. It was estimated that about 80% of global wastewater goes to the environment without any treatment. And most of wastewater streams contains several toxic organic micropollutants, including pharmaceuticals, organic dyes, pesticide, and other industrial chemicals. And these compounds have negative impact on ecosystem, for example, aquatic life, agriculture, as well as on health. However, due to their chemical complexity, the current existing wastewater treatment plants are not very effective to remove such compounds. In this regard, people have investigated various advanced oxidation processes to remove complex organic micropollutants from wastewater. The primary purpose of AOPs is to generate hydroxyl radical via various reaction pathways. The hydroxyl radical is one of the powerful non-selective chemical oxidant that can react with most of organic micropollutants in liquid. In my study, I used cold atmospheric plasma jet, which is chemical and catalyst-free advanced oxidation process to remove complex organic contaminants from water. So let's talk about how we define plasma. So as we know, a solid, liquid, gas, all three are well-recognized state of matter. Plasma is the fourth state of matter. It is ionized gas and mixture of electrons, neutral species, positively and negatively charged ions, and so on. So lightning and northern light, these are some common example of naturally occurring plasma. Plasma classified as thermal or non-thermal based on relative temperatures of electrons, ions, and neutron molecules. Thermal plasma concerned with high temperature in which electrons can achieve the same temperature as other species within the system. On the other hand, non-thermal plasma concerned with low temperature. So non-thermal plasma, also called cold plasma, is a source of high energetic electrons and bunch of reactive species at ambient condition. That's why we call cold atmospheric plasma, that can be generated at atmospheric pressure and close to room temperature. So cold atmospheric plasma is gaining a phenomenal attention in recent years for very promising applications. For example, water treatment, agriculture, medicine, in microelectronic industries, and so on. So now let's talk about plasma liquid interaction and related chemistry. So when the plasma, atmospheric pressure plasma jet interact with liquid, can generate several reactive species in the gaseous phase, at the plasma liquid interface, as well as in the bulk liquid phase. And these reactive species have a higher oxidation potential. They can react with many organic contaminants in liquid. So the fundamental limitation, the fundamental downside of at 1 atmospheric pressure plasma jet, the plasma volume is quite small, indicating that there will be problem to treat large surface of polluted water. So it is demonstrated one of the research gaps. So in this regard, the objective of my thesis, the goal of my thesis was to design, develop, and characterize a new plasma source that provides significant interaction between plasma and contaminants. So the entire objective is graphically represented here. The first aim was to develop a plasma source and generation of plasma in contact with liquid. After that, a detailed electrical and optical characterization of plasma system. Then the use of cold atmospheric plasma to treat a number of organic contaminants while consideration of several experimental parameters. Final step includes characterizing plasma treated samples by using several analytical techniques. So the first phase of experiments, like preliminary experiments, were conducted by using the existing plasma source known as 1-pin atmospheric pressure plasma jet. So here we can see the plasma is interacting with liquid. So the argon plasma was generated. Generated at the liquid surface in contact with ambient air by using a high voltage RF power supply. The various electrical probes were used for electrical diagnostics. The optical characterization was carried out by means of optical emission spectroscopy and ICCD camera. And the plasma treated samples were analyzed by using several analytical probes. A full electrical characterization was done in order to determine the working voltage, current, and power deposition to the plasma system. The recorded voltage and current waveform during the treatment is shown here. The voltage and current characteristics of a plasma system were investigated in two different circuits. One where high voltage was applied, one at the source where high voltage was applied, second one in the circuit where plasma was in contact with liquid. So the voltage and current characteristic studied in two different modes when there was no plasma and when the plasma was ignited. These following equations were used in order to calculate the power deposition to the plasma system. In our case, the more important was to calculate the power in the circuit where plasma was in contact with liquid. That was about 5 to 11 watts. The optical emission spectroscopy was used to study the gaseous phase of plasma chemistry to identify various reactive species within the plasma discharge. So OES spectra confirmed several reactive oxygen and nitrogen-based species, including hydroxyl radical and atomic oxygen. These are very critical reactive oxygen species in water decontamination. So it was also observed that when the input power was increased, the emission intensity of these reactive species were enhanced. The optical characterization of plasma was also carried out by using via ICCD imaging. The ICCD camera, in combination with UV lens, was placed in perpendicular to the plasma jet to capture the specially resolved plasma emission. The captured and processed ICCD image is shown here with the emission from whole active plasma. This blue line representing the liquid surface. The imaging was carried out with various filters. So several band pass filters were placed in front of the camera to detect the specially resolved emission from various reactive species. Those were identified during OES measurement. So each filter had bandwidth of around 10 nanometers. So for example, 310 nanometer filter was used to detect OH emission. So since it has 10 nanometer of bandwidth, excited nitrogen can also be recognized. So these are the images captured with ICCD camera with filters. First image is from OH and excited N2. So it can be seen that the brighter intensity of OH and excited N2 appeared in all along the jet. Excited atomic oxygen and excited argon were noticed in the plasma jet, but the higher intensity was at the downstream of the plasma jet. That means close to the liquid surface. Molecular nitrogen was also appeared. H alpha, on the other hand, was detected in the plasma jet with lower intensity. So these images indicated the presence of reactive species within the plasma discharge. So several organic contaminants were selected and treated with plasma source. Each compound had different characteristics. So these compounds were selected to check the performance of plasma source, which means how plasma will deal with how plasma source will deal and destroy these compounds. So for example, these compounds, for example, acid bull 25 dye, which is used in textile industry, diclofenac, which is pharmaceutical, and these compounds have been frequently detected in the environment, even though in Danube River as well. So third one is parachlorobenzoic acid, which is one of industrial chemical. So here I plotted the degradation of each compound as function of plasma treatment time. The treatment was carried out at 2 different initial concentrations and a treatment volume of 5 ml and the power deposition about 10 watts. So the first 2 compounds degraded in a very identical manner. More than 50% removal was observed in both cases within 3 minutes of plasma treatment time. So after 10 minutes of plasma treatment, there was almost complete removal. And the degradation was following the first order of kinetics. Diclofenac, on the other hand, degraded very rapidly. Here we can see that the almost complete removal was observed within 1 minute of plasma treatment time. Overall, the faster degradation was occurred with diclofenac, then then PCBA. Another approach was used where polluted water was treated. Polluted water such as dye containing solution was treated indirectly by using plasma activated water. So in the direct treatment where the plasma can be directly contact with polluted water, and bunch of these reactive species can contribute to the degradation of pollutant. On the other hand, in indirect treatment, the first plasma activated water was prepared and then it was mixed with polluted water. Plasma activated water contains various reactive oxygen and nitrogen based species such as hydrogen peroxide, nitrate, nitrite, and other reactive species. And plasma activated water can also be stored for several days. So the aim of indirect treatment was to check the role of check the contribution of long-lived plasma-induced reactive species on the degradation of pollutant. So there are limited study carried out in the literature based on the treatment of polluted water by using plasma activated water. So the first plasma activated water was prepared and the concentration of the reactive oxygen and nitrogen based species were identified. Separate solutions were prepared based on the identified species in the plasma activated water. For example, hydrogen peroxide, nitrate, and nitrite. And these reagents were combined and also mixed together with dye pollutant. So the plasma containing dye solution and these chemical containing dye solution were analyzed for 70 days. After 70 days of periodic analysis, it was observed that the higher dye removal was observed when the plasma activated water was mixed with dye solution and then with hydrogen peroxide containing solution. There was no significant dye removal when the nitrate and nitrite were mixed with polluted water. So the higher removal associated with the plasma activated water when the plasma activated water was mixed with dye pollutant can be attributed due to the presence of other reactive species in plasma activated water that I did not identify. I mean, those were not identified. So these preliminary investigations gave a lot of information in order to build a new plasma source. So a new plasma source was built known as 3-pin atmospheric pressure plasma jet. And the plasma was ignited by using same RF high voltage power supply. The approach of increasing number of jet was to increase the mass transfer of reactive species within the liquid so that a bit larger volume of water can be treated. And although there was possibility to increase more number of jets, but the system was optimized with 3 jets. Again, a detailed electrical and optical characterization was carried out, and the plasma treated samples were analyzed by using various analytical techniques. So the electrical diagnostic was done, and the voltage and current characteristics were almost identical as one atmospheric pressure plasma jet. But there was a difference in the power deposition. I mean, it was like a little higher than 1 jet. ICCD imaging was also done for 3-pin atmospheric pressure plasma jet, but with filters. So these are the filters. These are the images captured with ICCD camera with filters. So OH and excited N2 observed in all three plasma jets, but with a higher intensity at the downstream of plasma jet and close to the liquid surface. Atomic oxygen and excited argon were appeared mostly in the middle of jet. H alpha detected in the downstream of the plasma jet, and molecular nitrogen was also noticed mostly with the lower intensity in all along the jets. And the significant emission occurred. The significant emission from argon, excited argon, occurred mostly at the top stream of the jet, close to the electrode tip. So again, all these images indicated the existence of reactive species in the plasma jet. A new plasma source, a plasma recirculation system was developed. I would like to show you a video. Okay. Yeah. So a plasma recirculation system was developed where the plasma was generated over the recirculated polluted liquid surface. So this technique was used to enhance the plasma liquid contact interface and mixing, so that more reactive species can interact with polluted water, contaminants, and a bit larger volume of water can be treated. So with this configuration, a quarter liter of polluted water was treated. So here I have plotted the degradation of each pollutant as function of plasma treatment time. The treatment was carried out at a treatment volume of 250 ml and the power deposition of about 9.5 watts, which is almost similar with one plasma jet. First two compound degraded slowly. Diclofenac, on the other hand, degraded very rapidly. So almost complete removal was observed within 30 minutes of plasma treatment. So as I pointed out in the previous slide, these first two compounds were degraded slowly while treatment by using one jet at a treatment volume of 5 ml. So the volume can be one factor for slow degradation. So overall, the faster removal was noticed with diclofenac, then di, and then PCBs. The energy yield was also calculated as milligram per kilowatt hour for 50% removal, which indicate the amount of energy required to remove milligram of pollutant from water by means of 1 kilowatt hour energy. So the higher energy yield was observed during the treatment of diclofenac by using three plasma jets, 3-pin plasma atmospheric pressure plasma jet, and the obtained energy yield was high. The obtained energy yield was about an order of magnitude higher than other pollutants. So higher energy yield was observed with diclofenac. So the energy yield was also compared with the existing plasma-based pharmaceutical treatment data, and it was noticed that in our case, the energy yield was higher than the existing data, which is good. Some of plasma-treated diclofenac samples were also characterized by using Orbitrap LCMS in order to identify transformation products. About 7 transformation products were identified, and based on TPs, the possible degradation mechanism was proposed. So it was observed that when the plasma-induced reactive species interacted with diclofenac pollutant, diclofenac underwent through various reaction mechanism. For example, hydroxylation of aromatic ring, dechlorination, CN cleavage, ring opening, and then further degradation. And the concentration of these pollutants, TPs, were decreased with plasma treatment time. And some of TPs were also identified in the literature. So it's time to summarize and conclude my presentation. So I use cold atmospheric plasma as a chemical and catalyst-free approach for water treatment to remove organic micropollutant from water. A new plasma source was built having 3 jets to enhance the plasma liquid contact interface. A detailed electrical characterization was performed in order to calculate the working voltage, current, and power deposition through the plasma system. The optical diagnostics was performed to identify the reactive species within the plasma discharge. Several pollutants were selected and treated with plasma, and the highest removal was observed with diclofenac. Another, I attempted to treat polluted water by using plasma activated water, which is a new approach that can be used to treat contaminated water that being used for agriculture. Some of plasma treated diclofenac samples were identified, characterized by using Orbitrap LCMS in order to identify the transformation products. Overall, cold atmospheric plasma can be used to remove complex organic contaminants from water. The plasma recirculation system provides plasma recirculation system enhance the interaction between plasma reactive species and the contaminant, which fulfill one of research gap while providing a hope for scalability. Now I would like to give some future research remarks. For example, the plasma treatment of different water matrix, for example, a mixture of different pollutants and real wastewater samples, treatment of more number of pollutants. For example, in my case, I choose few pollutants. So treatment of more number of pollutants with having different characteristics. So for example, surfactants that can be efficiently removed by plasma. I use argon as a working gas. So argon can be blended with air so that the whole system can be more approachable. And accordingly, a new power supply will be needed in order to ignite air plasma, since air has higher breakdown potential. I used 3-pin atmospheric pressure plasma jet, but there was possibility to increase more number of jets in order to enhance the plasma liquid contact. Maybe another plasma source geometry can be proposed to enhance the plasma liquid contact interface, as well as to increase the mass transfer of reactive species in the liquid. A number of lab scale experiments and pilot scale experiments will be required in order to scale the plasma device for large scale water treatment. And then comparison with other existing AOPs. So maybe plasma source can also be combined with renewable energy sources, so that overall overall process can be more affordable. So during my PhD, I published 3 papers in 3 different journals. I have attended several conferences where I presented my PhD work. I have also attended several scientific events where I discussed about my PhD work. I wrote more than 5 blog articles. Most of them were related to my PhD work. These blog articles can be found at Novelty's project website. So first, I would like to acknowledge Novelty's project. Novelty has received the funding from Horizon 2020, MSCA and ITN. Many thanks to Dr. Nevena Puech for accepting me as a research scholar at IPB within Novelty's project. I would like to thank my PhD supervisors, Professor Dragan and Dr. Wolfgang, for their guidance. I'm so thankful to committee members for their valuable suggestions and comments in order to improve the quality of my thesis. I want to thank my lab colleagues, Dr. Nicolas Cora and others, for their support during my PhD. I think most of them are here. except Dr. Dan. He's attending online. So many thanks to Dr. Susanna, Dr. Elizabeth, and Dr. Maria Jose and Nicoleta for their support during the analysis of plasma-treated liquid samples. I want to thank Juliana for her support to deal with all the bureaucracy at IPB. Many thanks to Professor Nevenka Rice and Barbara for their support to deal with all the paperwork at TMF. I would like to acknowledge IPP and ICRA friends and Novelties team. And I would like to mention and thanks to other friends in Belgrade, like from Serbian Cricket Association and Panna team, for including me in outreach activities. Thank you for your attention.
Do we continue right away, or do you want?
Maybe I'll drink little water then.
Yeah. Okay. I suppose it would be prudent to continue in English for the benefit of the candidate. It wouldn't be fair to ask him questions in Serbian. Before going into questions, I would just like to state that Amit arrived to Belgrade shortly before the COVID lockdown. And during all of this time, he was most of the time in Belgrade, sometime in Spain. And he took advantage of every moment when Institute was open to go and do measurements and do some work. And not only that, but he joined a cricket club and made a major contribution to cricket in Serbia. So not surprisingly, the tallest building in Belgrade is shaped like a cricket bat. Check it out. So anyway, I would like to give podium questions to Professor Ivona Radovic, full professor at University of Belgrade, FTM. Please.
Thank you very much. Good afternoon, Amit. Good afternoon to our committee members abroad and to all our guests. Welcome to our faculty. First of all, as I have the opportunity to open this Q&A part of this defense, I would like to say that I'm really pleased and also honored to participate in this distinguished committee and to thank Professor Povrenović for giving me this opportunity. As a chemical engineer, actually cold plasma is not my field of expertise, so it was something new for me. But as the thesis is really very well organized, planned, structured, and very thoroughly and comprehensively written, I really enjoyed in reading it. So I have to congratulate to you, but also to your supervisors for really good job. So congratulate to you and all the supervisors. As I said, as a chemical engineer, I would like to move, more from the research and the laboratory scale to industry, I can say, the level. If you had a chance, so my questions are mostly related to that field. I prepared a lot, but don't worry, I will ask just one or maybe two questions. So it's very interesting topic, and we can see that it is the state of the art in wastewater treatment. So I hope that will also have its industrial application, and it will be future very soon. If you had the chance to move from this laboratory to move to the scaling up of the whole process, how would you proceed? I will maybe explain it. So would you go, for example, for a batch system or for a continuous system? Then would you choose discharge above liquid in the bubbles, or you also investigated the possibility of circulating of the whole system? So what would you focus on mainly? And can you apply the same approach for all the chemicals that you studied in your thesis? So if you can elaborate
Okay, so
it.
first I would like to answer from this question that you asked about the batch scale and recirculation. That will depend on actually the amount of polluted water and the types of contaminant. For example, as you have seen, that diclofenac was degrading very rapidly. So I don't think so. If we have a large, how to say, if we're going to put more number of needles, we can create large interaction. So maybe we will not need that like batch type. Maybe we can treat that compound with just how to say, in continuous mode. But there are some compound or complex. So in those cases, maybe pilot this batch mode treatment will be required. So again, I mean, at small scale, yeah, it depends. Yeah.
But what would you do also to enhance maybe the mass transfer?
Yeah. So there are.
Would you use this above liquid or
okay, I will go. So in my case, okay, plasma was generated in the gaseous phase over the liquid surface. So this is the good way, I mean, to generate plasma in gaseous phase in comparison to inside liquid, because it requires less energy, less voltage in order to ignite plasma in gaseous phase. So energy point of view, let's say.
Okay.
And as you said, as you asked that to increase the mass transfers, there are two technical ways. For example, one is, okay, change the geometry of plasma source, somehow increase the enhancement of the plasma induced reactive species and contaminants, and maybe change the flow direction, maybe somehow make flow over the thin film something, so that more reactive species can transfer. There are some ways to enhance. Yeah.
But do you think that it would be possible also to combine some well-known processes with this plasma? Is it possible also maybe to enhance the process? You also talked to me about the more effective would be the process where you have the effluent on the surface. So can you in some way enhance?
Yes, for example, for those compounds like surfactant, like PFAS, let's say. I mean, I have not treated PFAS, but one of my colleagues, he treated PFAS compounds. So for those compounds, like those compounds can be efficiently removed by plasma, because I'm generating plasma over the liquid surface where bunch of reactive, short-lived reactive species are presented. So those reactive, I mean, surfactant have tendency to enrich over the surface of liquid. So they can react and they can be degraded very rapidly. Okay. So there are literature, and one of my colleagues, he also investigated. She also used one of my plasma source to treat PFAS compounds. So it was.
And is there any industrial application in the world, or some pilot plants?
There are some people. They are, I mean, they have scaled up their plasma source, not for very high scale, but for like they're treating polluted pharmaceutical hospital wastewater. They are trying to remove pharmaceuticals, and some there is one group in US. They are treating PFAS compound by using plasma for US. Air Force. So yeah, there are, but small scale, not very large scale. Yeah.
So the technological readiness level is maybe
it's 4 and 5. 4 and 5. Yeah, 4 and 5. 4 and 5 because it is proved in the lab, and some people they have also scaled up for pilot scale, let's say.
So are you planning in this lab to do it to go further? Do you have some knowledge about that, or you will investigate on this experimental level in the laboratory?
Can you
here in the lab at the institute?
Maybe someone will do that in future. Now we have a lot of we have done fundamental investigations, so we have a lot of data. So if someone is going to continue, it's good. Huh?
Okay. Thank you very much. I would like to let other to ask questions also. Thank you. I'm really satisfied.
Okay, let's proceed. Now we will take questions from Zdenko Mahala. Good morning, or is it good afternoon, Zdenko? Thank you for joining us.
Good afternoon, everybody. I hope you can hear me well.
Yes.
Yeah, perfect. My excuses for missing the start because I had some connection problem at the beginning, but then I followed since the beginning, almost since the beginning, the presentation. So first of all, let me, I would like to appreciate this thesis of Amit Kumar, which I read sometimes a few months back, but it was a really interesting reading. It was really well organized, clearly written at high English standard, and there was a nice logical thread. So it was kind of interesting reading, like a novel almost, scientifically. And I also really appreciated very focused and attractive presentation that Amir just showed us here. Sorry, maybe I should have introduced myself a little bit. So my background is plasma physics, and I work a lot in research on plasma liquid interactions, plasma activated liquids, and some applications in medicine, in agriculture, and also diagnostics of plasmas and plasmas with liquids. So this was a topic which really was in my field, although I'm not a chemist, so I couldn't really go perfectly deeply into the chemical part of it, but I appreciated that there were 4 different chemicals, this AB25, diclofenac, PCBA, and DR1, which were tested and compared. And one of them, I think, was diclofenac. Even in the end, some mechanisms of hydroxylation and other reaction pathways were analyzed in depth. So that was really that's really very it contributes a lot to the general knowledge or what happens to these chemicals upon plasma treatment. And also, as Amit showed us, so he used a single plasma jet and a triple plasma jet in direct treatment, as I understood, and also then indirect treatment, just treating water and then applying this plasma activated water to the decontamination of these compounds. So much kind of a general overview. Maybe I can have some questions now. I'm not sure how many questions can I have, because I have many, but maybe I can just select some of them. So maybe on the optical emission spectroscopy that you showed us, which was one part of the thesis, I didn't quite get it. I saw on your presentation that you used an optical fiber, but there was no focusing system with lenses or mirrors. So it was just a fiber kind of pointing to some height or some level of the plasma jet. My question is, where was the spectra taken from? Close to the liquid surface or farther up, or closer to the plasma jet, where as it goes out from the tube? Where was it taken from? Or did you perhaps do some profiling?
Okay, so I understand your question. So there was a lens connected with probe. So the probe was placed in such a way that the emission from whole active plasma can be captured. So I have done with several distances. So let's say in case of pin jet, the emission was mostly from whole active plasma. I mean, tip to close to liquid surface.
Mm-hmm. So was there any difference, for example, in OH spectra, if you focus close to the liquid surface and if you focused higher up at the effluent of the plasma jet from the tube? Was there, or you didn't try these things? I'm not sure now.
Actually, I tried, but the intensity was varying. Intensity of each emission lines or bands were varying. Whenever I was putting that probe close to the plasma or taking away from the plasma, so the relative intensities were changing as whenever I was moving the probe.
Okay, I see. Yeah. I was just curious if you could somehow maybe track some of the species, especially those relevant to the liquid, such as OH radicals, how far they go, or if they are evenly distributed along the jet or not. Well, but anyway, this was not the focus of your work. Of course, you've done many different other things. I was just curious. Okay, and then also when I looked a little bit with detail on your ICCD images of the triple plasma jet, and there were these three, and as I understood, this triple plasma jet, it was powered by the same power supply. So were the three jets really identical in terms of physical parameters? Because the ICCD images show some differences, but maybe they were taken, I'm not sure how were they taken. You took a snapshot of all three at once, or they are just put one close to the other? Because I see some little differences in these images.
Okay, actually, the camera was placed in such a way that all, perpendicular to the plasma jet, so that the emission from all these three jets can be captured. But I also done the analysis of this middle jet. I focused the camera only on middle jets for detailed investigation, but I did not put the results here. I have in backup slides. I can show.
Yeah. So were there any physical differences between jet 1, 2, 3 when they were operated at the same time, or were they completely identical?
Actually, there was a difference. For example, the emission, the plasma, there was different plasma emission in each jet.
But then later you showed us that when you use this triple plasma jet for one of the pollutants, I forgot which one, it was more energetically efficient than using a single jet. So this is a good way to maybe kind of scale up, or indication that it could be scaled up this way. I was just how many such jets could you power from the same power supply?
I tested up to just 3 jets. Although there were possibility, but I stuck with 3 jets only.
Okay. Well, again, it's just curiosity. If you can, perhaps with a bigger power supply, more powerful, you could scale up more. And you mentioned that the direction several times, so that could be interesting. Because otherwise, of course, this is as you also admitted, this is a relatively small, localized treatment of relatively small volume. So it's if to be used in industrial scale, it would have to be scaled up quite dramatically. Okay, then I noticed that the temperature, when you use the single plasma jet, the temperature when you treated 5 milliliters increased up to 64 degrees or something like that. It was a table in your thesis, I forgot the number of the table. Nevertheless, when you use the triple, it was not so, because probably the volume was larger, I guess.
Yes.
So my question is, if the temperature increased as high as 60 degrees, is there an effect of the temperature itself on the decomposition of these chemical compounds, or does it contribute, and how does it contribute? Did you do some comparative measurements of just heating the mixture and looking what happens?
Okay, so the temperature was increasing only when during the treatment of 5 ml of polluted water by 1 jet. So,
yes.
but on the other hand, in case of 3 jets, there was temperature was not increasing. It's going like maximum 29 or 30 degrees Celsius due to heat transfer and a lot of convection heat transfer. So I did not do any investigation the role of temperature on degradation. So, but increasing temperature, there are some literature where I studied, so they increase the liquid to certain temperature. They were trying to see how temperature will influence on degradation. And they observed temperature was playing important role to enhance the degradation of pollutants. But okay, at very small scale, when the volume was very small, like 5 ml.
Okay, but okay, thank you very much. And then with the chemical AB25, that you investigated the gap distance influence, and it was quite interesting that with the gap distance, the removal increased and then decreased. It was, I think, figure 517 of the thesis. And I wonder why that happened. Why is this middle gap distance the optimum to get the highest decontamination? Yeah, if you have any idea.
I would say, I mean, when the highest highest removal was observed when the discharge gap was 10 millimeter, and then when I was increasing, then the removal efficiency was going down. So maybe
why was it lower for shorter gap distance? Yeah, it's clear that when you then make it longer, the efficiency would decrease. But why the other side would go down?
Maybe some plasma induced reactive species were quenched. That could be, I mean, one reason. I also tried to find in the literature. So I found some of information. So one reason is could be the quenching effect of reactive species.
Quenching by
quenching by some of like reactive species. I mean, some of species when the argon plasma is interacting with atmospheric air.
Yeah, but I don't quite understand why would be they quenched for short distance and not for longer distance, when for longer distance there's a longer time to reach the surface and they would not be quenched, and for short distance they would be quenched. I'm not quite getting this, but maybe the plasma parameters and the power for a shorter gap distance would not develop as much as in this if we compare this 0.5 and 1 centimeter. I'm not sure, I'm just guessing, but this quenching explanation, I don't quite get it. Okay, never mind. And then there was an interesting result, really interesting and I really appreciate all the study because there's a lot of work behind that. And on your figure 518, you showed, and even in the presentation, you showed the effect of plasma activated water, which was the fastest degradation, and then different chemicals like nitrites, nitrates, hydrogen peroxide, which induce some level of degradation, but much weaker. And then you compared the direct, and then when you did the mixture of these different components, it was not as good as the real plasma activated water. Can you show that picture? You have shown it in the presentation. It's also in you.
Yeah, here it is.
Yeah, yes. So why this case of when you put all of them together, the red squares, and compare with real plasma activated water, which is the blue half moons, whatever. I cannot see it really well from here. Why this plasma activated water is so much better than when you add all the chemicals together? Yeah, now I see it better. Yes.
So maybe there are some other reactive species like were available in the plasma activated water that I did not identify or characterize. Like for example, peroxynitrous acid, that is also one of the powerful chemical oxidant that can be generated in plasma activated water. So I just identified H2O2, hydrogen peroxide, nitrate, and nitrite. So there could be more stuff. I mean, more oxidants. They can play important role for the degradation or higher degradation.
Yeah. Okay. And so this was all the indirect treatment. So the plasma activated water, and then you added that to the chemicals.
Yes.
And if you compare this to the direct treatment, so the chemical is already there when it's treated, which one turns out better? Are they comparable?
Yeah, I did some separate experiments where I treated this dye solution directly with plasma, and then I kept again for several days. I was analyzing. The difference was not more significant. because when the pollutant was there, those plasma-induced reactive species interacted with pollutant. So they were not, I mean, maybe these H2O2, NO2, H2O2, for example, was not going inside the liquid when the pollutant were presented in the liquid.
Ah, okay. So that
Yes.
so the transport of species was somehow influenced by the presence of the pollutant. This is what you mean?
The transfer, I mean,
The transport of, for example, hydrogen peroxide or other reactive species from plasma, when it goes just into pure water, and when it goes into the water with the pollutant, it's not the same. Is that
not the same? Yeah.
Ah, that's very interesting.
Yeah.
And that's really important also as a fundamental study in this sense to show that it's really, it's not the same, even if you use the same plasma source, same parameters, same everything, but if you treat different types of solutions, it may have different effects.
And I would like to I would like to mention that I also put this dye solution at lower pH. I mean, I just decreased the pH, and I kept for several days, and I was trying to see if dye can be degraded itself at low pH. So there was no degradation at all.
Okay. Okay. Thank you. Thank you. Good remark. And maybe the last question related to this. So you treated single types of pollutants, these 4 types. Have you perhaps tried their mixture or the mixture of several pollutants at the same time, which is more representative of a real life contaminated water? And is there any, or if you did that, was there any influence of one pollutant to the effect of degradation of the other? Did they influence each other?
I haven't done that. I mean, I was treating just individually. Yeah. So that can be like future work. I mean,
yeah, sure. That could be something interesting because they can really influence each other. I think that's my guess. Of course, I haven't done these experiments on your compounds. Okay, thank you very much for your answers. And
Thank you,
yes.
No, just saying thank you for the questions.
Oh, okay. Yeah. Am I supposed to say some conclusions or not? Not at this moment. I'm not sure how this is organized.
Not at this moment. We will have a separate session of the thesis committee afterwards. The next series of questions will come from Sara Rodriguez Moses, a senior research associate of the University of Girona in Spain. Please.
Yeah, hello, good morning, and thank you, Amit, for your presentation, and also your directors and supervisors for inviting me to be here. I am chemist, I'm an analytical chemist, so my questions will be general questions, but also some specific to micropollutants, I would say. So yeah, congratulations for the presentation. It was pretty clear to me. And I acknowledge and I appreciate that you structure and this outline and to give the objectives and some of them. It was, in my opinion, more clear today than in the thesis document itself. So I appreciate that and I thank you for that. And it was quick and you stick to the quick or fast and you stick to the point. So I appreciate that too. So congratulations for that. So I have a couple of questions. My first question is about the selection of the targets or the model pollutants that you were choosing to launch or to perform your analysis. Because when I was reading your thesis, you were paying attention to pesticides, for instance, pharmaceuticals and some dyes, okay? But in the end, you chose these 4 compounds. None of them was a pesticide. And I was wondering what was the rationale, or what were the reasons behind the selection, and what happened with the pesticides. You answered already about the perfluorinated compounds, that because it's obvious that they are like important ones, but some other people were analyzing or were targeting them. So what was the criteria to select those compounds in particular? Is it based on structure and recalcitrance of these compounds of the or the future applications? So what can you tell me about it?
Yeah, so these compounds were selected. I mean, each compound has a different characteristics. I mean, different solubility, different reactivity. So the main aim was to see how plasma source will behave during the treatment. So we wanted to check the plasma treatment efficiency. I mean, how plasma will destroy these compounds. So we stuck with 3-4 compounds. I mean, you are mentioning the pesticide. I mean, I did not use pesticides during the treatment. So for example, these first 2 compounds are frequently commercially used in industry, like acid blue 25 dye, which is highly consumable in textile industry, and diclofenac is one of, I mean, famous pharmaceutical. So these compounds are also frequently detected in the water bodies. So the third one is just, I mean, this is also used to make pharmaceutical. I mean, one of the industrial chemical.
Yeah, no, I was kind of surprised because you were devoting some effort in the introduction to describe pesticides, and there was a table talking about the applications. I mean, I think that there are 3 tables, one for pharmaceuticals, one for dyes, and another one for pesticides. Like, and you devoted a lot of effort in doing a literature review on those, you know, you selected those families, but in the end, you didn't target any pesticide. That was the reason of my question.
I just put in the introduction, like these compounds are identified in the natural water bodies, like in the environment. I mean, that was just in the introduction.
Yeah.
Yeah.
Okay. Yeah. But you also said in the thesis, but also today, like you were talking about future application or potential applications of the technology, and you were mentioning the use in agricultural effluents, and I didn't get what you were actually meaning, like a runoff from agricultural land, or just the irrigation water? What did you mean by applications?
Okay, so for example, the water is being used for irrigation, like
Okay.
containing a lot of pesticide or other compounds, for example. So those compounds, like those water can be treated by using like, let's say, plasma activated water or directly with plasma. So the plasma will destroy those compounds, as well as will provide some these long-lived reactive species or some nutrients, for example, nitrate, nitrite. Those are like, they can work as a fertilizer for plants.
Okay.
So there will be 2 effects. One is,
okay,
plasma can degrade compounds, and other effect can be this those species, like those nutrients can work as a fertilizer for plants.
Okay, understood. Yeah. So in that sense, you just said like the contaminants are destroyed. Okay? Like it's like a strong statement, or because you actually studied the generation of transformation products of organic pollutants. No, so when you talk about destroying contaminants, I guess that you are meaning like the removal of the like main compound or the target compound, but actually they are not totally destroyed or mineralized, no, but they are generating these transformation products. So I guess that you agree with me about
Yes, I mean, I've done this analysis only for diclofenac and identified those transformation products, but those the concentration of those products were decreasing with treatment time. So for longer exposure of plasma, those transformation products can also be eliminated from water. I mean, I have not done for these other compounds like dye and this PCBA, but I found some of information in the literature. They have done some toxicological test. after the plasma treatment, they also claim that, I mean, there was no major adverse influence on bacteria or something, or some.
Yeah, that was actually my next question. It was about the toxicity issues, I would say. Yeah, I mean, I think that in the literature you can find all kind of results. Like some people are reporting increase in toxicity and some others a decrease. My question about this toxicity issue is that if you, I haven't read it in the thesis, so I see. So you didn't perform these experiments or evaluation, but did you think about it, like checking? Or did you consider to use, I don't know, a general tox assay to evaluate the toxicity of the treated water, the treated contaminated water? Or this is in the case of actual wet experiments, but you know that there are also some models available to predict toxicity. So I was wondering if you explored that possibility of checking the toxicity of the transformation products that you generated in the experiments with diclofenac.
Yeah, I mean, I have not done any toxicological test, but I should have mentioned that in future work also. I mean, I did not put, but it should be done. I mean, before scaling up the device. Yeah.
I think it's a important thing to tackle, yeah, to deal with, even to pay attention to it, even if you didn't have the time to do everything, of course. But I think it's an important issue to check the toxicity. And yeah, I think that and did you also, that will be my last question, I think. I mean, you didn't do the mixture, the treatment of a mixture of compounds or whatever, but did you think in spiking like real water, either industrial or urban treated wastewater or whatever? Did you think, or did you even try to spike one, just to check the matrix effect on the plasma performance?
I haven't checked. I mean, I haven't treated different water metrics. I was just treating individually to see the performance of plasma source. So, I mean, that's why I put in the future work, like that should also be done with different compounds, treatment of real wastewater samples. If there are a bunch of compounds, then we'll see how those reactive species are interactive with pollutants to check the total organic carbon, then mineralization and other things. Yeah. Those things are also important. Yeah,
future work and you had bad luck with the actual or the past situation with COVID and so on. So you working under those challenging conditions, must be. So congratulations to being here after all of this. So that's it on my side. Thank you.
Thanks.
Thank you. Now we'll proceed to Jelena Rađenović. We'll ask the next round of questions.
Thank you. So good day, everyone. Dobar dan. I don't know, Amit, if they call you Amit or Amit. Do they decline it in Serbia or? Amit, no?
Amit.
Okay. So, well, first of all, congratulations. I think you did a great job with the thesis. The written language is really good. I think you also made a nice presentation. And as my colleague said before me, I think it's really great that you managed to do so much work during COVID and in a project that is pretty challenging. You know, I mean, it's not easy for students to do a thesis in 3 years, even in one institution, and you had to change. I think that's really great. And so, I have some questions. I mean, it's not easy to work on a technology that is really so new to water treatment field. So I understand that you had you only scratched the surface, right, on several issues. You don't have complete answers, and it will for sure serve for future developments of plasma-based water treatment. you know, some of the results that you were getting were really interesting. And so I'm not going to question so much the choice of your contaminants in terms of whatever environmental relevance. You know, there is, I was also a little bit confused with what Sarah mentioned. You know, a lot of focus on pesticides, and then the choice was a little bit surprising. But I'm just more wondering about the results we got for these compounds, because you got so like fast disappearance, fast oxidation, I assume, of diclofenac, and then for parachlorobenzoic acid and this dye, the results were different. So how do you interpret that? So what do you think would be the main oxidant species in your system?
Like reactive oxygen species, for example, hydroxyl radical, atomic oxygen, there could be peroxide radicals, superoxide ions. I mean, for example, diclofenac is not only reactive with OH, it is also reactive with other oxidants having lower oxidation potential. So that could be one reason, like diclofenac reacted with.
But sorry to interrupt, but AB25, why wouldn't that react as fast, let's say?
I mean, it is reactive. It is reactive with chemical, those reactive species. But the one reason could be, see, these byproducts. For example, the byproducts after the degradation of dye may be quenching the fresh, this new reactive species generated by plasma. So that could be one reason. So, like after the degradation, there might be some byproducts. They can react or quenched plasma-induced reactive species, even in case of PCB also.
Okay, but have you compared, for example, the reported second-order rate constants, like the molecular rate constants of these contaminants with like ozone, hydroxyl radical, whatever? Did you make this comparison to see?
No, I haven't done that comparison, like this kinetic.
Okay, well,
yeah.
Maybe it would be useful. Okay, for future work. Okay. Did you measure ozone in your plasma?
No, I did not measure ozone, but there was ozone. Like I can feel the smell of ozone.
Okay, well, so you know, for the figure that was mentioned just before, this figure 518, where you compare, you add peroxide with nitrate, nitrizone, you know, maybe, you know, that's not a complete figure in that sense. Like maybe you do have an effect of ozone much more than, for example, peroxide.
Yeah, I mean, ozone, I mean, I did not identify or measure ozone, but literature, I mean, some people, they have done the analysis to quantify ozone in the liquid phase.
So ozone would probably be there then.
Yeah, yes.
Okay. And then I was wondering if you managed to compare, I mean, I know that your system is very simple, and you know it's very difficult to talk about any kind of energy consumption for lab scale systems like that. Okay, but like, do you have any indication how it would compare with other technologies? Like,
I calculated the energy yield like in terms of milligram per kilowatt hour as well as in kilowatt hour per meter cube, as you mentioned when you were going through my thesis. So after that I calculated. So I found that the highest, in the best case, it was like 8 kilowatt hour per meter cube of energy consumption. That was comparable with existing AOPs.
Okay.
Yeah,
that's good. Okay. And then, okay, I'm just going to have one more question because I think we don't have much time. This temperature increase, which I also noted, I wonder, you know, that's maybe, I don't know, a little bit of a challenge for upscaling of this technology. Do you think this can be minimized?
Actually, one challenge is this generation of cold plasma at ambient condition. So, but that limitation can be overcome by using some noble gases, for example, argon. I know it is expensive, but maybe argon can be recirculated and can be reused. So,
but so the water is not going to heat up with that?
No. Water is not going to heat up. You're saying heating.
Heating.
No. Water is not going to be heated up if the initial volume is a bit higher and water is recirculating. So I calculated the temperature. It was not going like
Okay.
more than 29 degrees Celsius, even though for longer period of time.
Okay. Okay, that's all. Well, congrats and all the best, of course, for your future endeavors.
Thanks.
Thank you very
Thank you.
much. Now let's proceed to Professor Vladimir Pavicevic from our host faculty. Please.
Good afternoon to everybody, to distinguished members of committee, and to Amit, of course. I'm not familiar with plasma technology, so I'll be very quick in my questions. Let me learn something new. Can you tell me why did the distinguished group of institute choose atmospheric pressure plasma jet method instead of the electric barrier discharge, corona discharge, gliding arc, or something like that?
So I would say like it is easy to ignite plasma with that configuration because it has needle sharp edge, so high intensity of electric field can be generated over the tip of electrode, so it can be easily ignite the plasma.
Okay.
With lower voltage.
Please compare energy yields of your method with other advanced oxidation processes.
Yeah, so as I mentioned that I calculated energy yield in terms of kilowatt hour per meter cube. So the
Ozone, especially ozone,
okay.
Photocatalysis.
Okay, so as I mentioned that 8 kilowatt hour per meter cube, that was the best in the best case. So that was compared.
Diclofenac.
Yeah, for diclofenac, yeah. So it was comparable with existing AOPs. That I don't know exactly about photocatalysis or something. So, but it was comparable. I mean, in some cases, even though it was higher, in some cases,
very similar.
Yeah, I mean, in some cases it was higher also. I mean, we achieved higher. I mean, good, yeah, than them.
And it's obvious that pharmaceutical and some parts of chemical industries are main perspective for this implementation in practice. And I think maybe printing industry and packaging industry is also very perspective because they have they are washing their cylinders or rolls for coating inner layers or outside layers of packaging and carbonate. So they have a very big problem with varnish or seal. Varnish or seal. They are not biodegradable and they have a extremely high chemical oxygen demand. So I think that will be very interesting field of interest for the future investigation.
Yes, I mean, since like plasma can generate like bunch of radicals and bunch of reactive species, they can, I mean, degrade complex compounds, even those are non-biodegradable. So for
they have a very small volumes, but high concentration, very high concentration. They are very complex organic compounds. So I think maybe some future research may answer to those problems.
Yeah.
Thank you.
Thank you. Okay, now it's my turn. I wanted to ask you, I mean, as a user of anti-inflammatory drugs, I wanted to ask you, is diclofenac really a major pollutant, or is it just used as a model or for convenience in this study?
So it is banned in some countries, like in India, Pakistan, and Bangladesh. Diclofenac is banned because of a lot of birds were dying. So it is
banned. Which application of diclofenac?
For painkiller.
Because if I am taking my pills, I'm not killing any birds. I can hardly walk. So
yeah, but at the end it is going in environment.
At the end, when I die, or
no, through fluid system.
You are alive.
Okay.
Through fluid system.
Okay, so it is a major pollutant.
Yeah.
Okay. Well, I apologize for my contribution. I do not feel diplomat, but something similar.
I'm eating as well here.
And using plasma will add price tag to agricultural products. Using plasma will not make agricultural products cheaper, only slightly more expensive. So one should look for application where actually usage of water is small and productivity is large, which actually spells hydroponics. So my question is your system, let's say a certain plant becomes legal in this country. It is legal around many states around the world, and people are growing it and using terraces or small areas and using hydroponics to grow it and artificial light for 24-hour. So I'm asking, is your source of activated water big enough for small-scale hydroponic production of actual something which can be sold at the market?
Okay, so you're saying hydroponic, that is plant does not require soil, no soil, right? I mean, just nutrients and water.
Water is just dripping droplet by droplet.
Okay, so
let's focus on usage of water. It's very small.
Okay, yeah, I mean, it can be effective for
your current source, is it how big production can you see with the current source?
With current source, I mean, I'm just treating a quarter liter of water. So, I mean, I don't think so. It's going to be large, but by increasing number of jets, maybe we can increase the interaction, like plasma liquid interaction, and we can treat more. That can be maybe for space exploration and all. Yeah.
That's a good application.
Yeah.
Okay. Well, what did you use to tell yourself I have reproducible plasma? Which diagnostics?
In diagnostics, I use optical emission spectroscopy and ICCD imaging.
Okay. Did you determine power?
Okay. I've done electrical characterization, detailed electrical characterization by using several probes. So, to understand how the voltage.
Make sure that it is reproducible.
Yes. Yeah. I have done like 2-3 set of experiments. Yeah.
Well, that's as far as I will go. Do you have custom here to extend questions to the audience? Supervisors? Okay. Well, I suggest that the committee goes to a separate location to elaborate on this presentation.
Tamo ćemo ih pozvati.
You are muted. Become
Preparation
okay.
and signing, which is that Amit Kumar has defended his thesis with grade excellent, and the rest will be decided at Girona University in days to come, and he will get another even further specification. So I would like to congratulate Amit and wish him success in his further career endeavors, whatever.
Thanks.
Congratulations.
I'm not sure if Nevena wants to say something.
Thanks a lot.
Yeah, I would like to thank our foreign members of the Thesis Defense Committee very much. As I told you over there, but I will repeat in front of the whole group, it sounded very much like Eurovision.
Come.
We need a few more members of the committee, and we will have the whole Eurovision for the next Thesis Defense. Thank you.
Amit, you have here ICRA people wanting to congratulate you, eh?
Yeah.
Can you hear me?
Yeah, I can hear you. Yeah.
Congratulations.
Thanks a lot.
Yeah, congratulations from me as well, Amit.
Thank you.
It was really great. I think the anecdote about the cricket team is a very good example for everything that relates to like how you're not only hardworking, intelligent, and high achieving in 3 years, achieving not only like to stand here today, but also having your publications already published. All PhD students know that is very challenging to achieve this in 3 years. But it was really also great, like to see how you integrate it in the different workspaces and in society as well. I mean, I still really memorize with pleasure the day we went sampling, basically into the mountains with like a really bad car. And all the anecdotes that you told me, and I really hope you enjoyed your PhD, and I would like to thank my co-supervisors from the University of Belgrade for these wonderful collaborations that we developed around your PhD.
Thanks a lot.
Congratulations also from me, and congratulations for surviving Serbia as a vegetarian. I think that's amazing. Tell me how you do it.
Congratulations also from my side, from Bratislava, and I wish you good success in your future. Yeah, congratulations.
Thanks.
Can I use the thesis now? Is it kind of a public? Can I distribute it to my students, for example, to my people group?
Yes, of course. Okay.
Yeah. Thank you.
Thanks.
Congratulations, Amit. I join my colleagues also in acknowledge your work and a lot of success in your future projects. Okay.
Yeah.
Good luck.
Okay. He actually already has a job in Germany that he already has started as well. So he's really quick.
Yeah, I'm going to join next week.
Okay, so please.
Okay, so people, we have some cocktail, so some snacks, so please join.
You are
Okay.
welcome to watch the cocktail.
Great to see also that there are quite a few people in the room.
will be vegan cocktail.
It's always nice if you have a bit of an audience.
Okay, enjoy your cocktail and enjoy the day.
Yeah.
Thank you for cooperating and inviting me. Thank you.
Thank you very much.
Okay, see you, Wolfgang, and see you, everybody. Goodbye.
See you, bye.
Bye.
So I'm also leaving unless that's
Bye bye.
there. It doesn't seem
Bye.
so. Bye.
Da, što njoj treba da se nosi. To trebate ti Petrović, oni će da postavljamo. Evo dva, vlada, kad piši što je sigurno sigurno.
Ne osjećaš se kad mi čukeva. Navikao sam se.
Ne, ovo je katalonski, ne znam. Crnogorski i srpski. Mislim, potpišite vi.
Ovo je moguće da je ocena.
To je moguće ocena, dobro.
Ne, tu se potpisuje.
Tu se mu zamenili. Vi ste sad ovdje, a pitat ćemo, potpišiti ovdje isto, da vidimo da li je.
Jeli, koliko spavaš noću? Ali kad dođu osvetnice, onda ćeš da uživaš.
Promijenio je dan, pa kako šta spava? Znači,
uvek spavaju kad ne treba.
ali već
Ali ti si najvažnija, to je upravo. Zna ko je Keva? Keva je number one.
izbog strane, ja bih
Može biti ovo ocene.
Gde ja da potpisujem?
Vi ste vokal. Vokal.
A ovdje ili ne?
Ne, tu izgleda da ide ocena, dok ne.